Okay, so let's move on to section 6 of chapter 6, talking a little bit more about bone remodeling, so bone breakdown, and bone deposition. So, about 5 to 7% of our bone mass is recycled every week. Our spongy bone is replaced about every 3 to 4 years, and our compact bone is replaced every 10 years. So, what's really astounding about these numbers is that we basically replace our entire skeleton every decade. Every 10 year we have new bone, basically. When we talk about bone remodeling, like I talked about before, it's made up of both bone deposition, which is accomplished by our osteoblast, and then bone reabsorption, which is accomplished by our osteoclast. And it occurs at the surfaces of both your periosteum and your endosteum there. Okay, bone reabsorption or bone breakdown, we've already learned that this is the function of the osteoclast. How do they do that? Remember, they have the ruffled borders, and they have the enzymes there to help break down the bone itself. So they go in and they dig these depressions or these grooves as they break down the matrix, and they secrete lysosomal enzymes and protons or hydrogen ions, and that is going to contribute to the acidity. So the acidity or an increase in hydrogen ion concentration is also going to help digest the matrix. We can also use the acidity to convert calcium salts to soluble forms, so we don't have a hard mineral salt there anymore. So osteoclasts, also can go in and phagocytize demineralized matrix and dead osteocytes, and then we can take those products that they just basically digested, send them across the cell, and release them out into the interstitial fluid and eventually our blood. So once reabsorption is complete, our osteoclasts actually undergo apoptosis. So once they're done, they basically die. So again, just as a recap, osteoclasts, yes, they're responsible for breaking down the bone. How do they do that? The enzymes that they secrete are acidic because they contain high amounts of hydrogen ions, and then they can go in and break it down inside, translocate it across the cell, release it into interstitial fluid, which is eventually going to empty into our blood. They die after they're done. So what's going to stimulate our osteoclasts to go in and break down bone is a hormone called PTH, or the parathyroid hormone. We talk about bone deposition. We know that is going to be accomplished by our osteoblasts. A couple things to think about is that what happens is the proteins in our osteoid are going to go and they're going to bind calcium. When they bind calcium, they're increasing the local concentration of calcium. And this increase in local concentration of calcium is going to stimulate the osteoblast to release these vesicles that are filled with alkaline phosphatase. And alkaline phosphatase is an enzyme that's going to cleave a phosphate off of osteoid proteins to increase the local concentration of phosphate. That process is not really important for you to memorize or understand. What's important is that you need both high levels of calcium and phosphate in order to make those calcium phosphate crystals that we call hydroxyapatites that we need in our bone. Okay, so control of remodeling. Let's revisit hormonal controls. I mentioned previously the parathyroid hormone. So remodeling, like I mentioned, occurs throughout our lifetime, almost continuously, but it's regulated by a couple things. We have some genetic factors and a couple control loops. So when we're talking about hormonal controls, these are basically going to help maintain calcium levels in our blood. So they use the negative feedback loop. So basically, once we have too much of the hormone produced, it's going to go back and inhibit the production of the hormone to begin with. So negative feedback loops help control the blood calcium levels. Calcium functions in many processes that are vital to our survival. So for example, our nerves can't fire, our muscles cannot contract if we don't have the correct calcium levels in our body. So you'll learn more about this when we hit the nervous system and the muscular system later in the semester. Again, I mentioned previously, our bones are basically our calcium storage bank. So 99% of our calcium is found in bone. We deposit excess calcium to our bones, and if we don't have enough calcium, we withdraw some calcium from our bones. Another important thing to keep in mind is that in order to absorb calcium in our intestines, we have to have the active form of vitamin D. So it doesn't matter if you take a calcium supplement that's 3,000 milligrams of calcium, you're going to basically pee it out if you do not have vitamin D there in your intestines ready to help absorb it across your intestinal membrane. So calcium is useless unless you have vitamin D. The other way we can control remodeling is response to mechanical stress, which we'll talk about Wolff's Law in a second. So let's talk a little bit more about hormonal controls. Parathyroid hormone is your major player here. Um, Basically, it only works to control the blood levels of calcium, not the actual strength of the skeleton itself. So we have to have very specific levels of calcium in our blood. If the levels drop too low or they drop too high, it's going to trigger something to happen. So when we talk about production of the parathyroid hormone, which is a hormone secreted by the parathyroid glands, it's going to be secreted in response to low blood calcium levels. So if we have low blood calcium levels, we want to increase the amount of calcium that we can find in our blood. So what is it going to activate? It's ultimately going to activate our osteoclast to go in and break down the bone so that it can break down the bone, send all of the calcium to our interstitial fluid and eventually our blood so that we can raise our blood calcium levels. So if we have low blood calcium, parathyroid hormone is going to go in and be released. It's going to stimulate the formation of osteoclasts so we can break bone down so we can retrieve the calcium and increase our blood calcium levels. This is in here. This again, rank L is not that important. Basically, what it's telling you is that the parathyroid hormone stimulates cells to release rank L. Rank L is what's actually going to stimulate the formation of osteoclasts. So the parathyroid hormone basically increases blood calcium levels, but indirectly because it has to go in and activate rank L or cause something else to release rank L in order to stimulate osteoclast formation. But again, what is it doing? Its whole function is ultimately to raise our blood calcium levels, and it was secreted in response to low blood calcium levels. It works off of a negative feedback loop. So when we get enough or high enough calcium levels in our blood where we want, it's going to go back and inhibit the parathyroid hormone from being secreted in the first place. Calcitonin has the opposite effect. So basically, if we have high blood calcium, calcitonin would come in. This is somewhat debatable. Some textbooks say that calcitonin basically goes in and can lower blood calcium by stimulating the deposition of calcium into your bone. Your textbook says this only occurs if you get high doses of calcitonin. But for the sake of this exam, you should recognize that calcitonin at high levels does function as an antagonist to the parathyroid hormone and that it helps lower blood calcium levels, where parathyroid helps increase blood calcium levels. Okay, so what are we talking about when we talk about calcium levels? Again, they're really, really important in order for us to contract our muscles and stimulate action potentials. So when I say hypocalcemia, that just means low levels of calcium. Hypercalcemia means high levels of calcium. So we're putting these terms in relation to the two hormones we just talked about. The parathyroid hormone would act in response to hypocalcium because its function is to increase blood calcium levels by stimulating osteoclast activity and bone reabsorption. Calcitonin would act in response to hypercalcemia, where we have too much calcium in our blood, so it's going to stimulate bone deposition of calcium. The other way that we can remodel our bones are in response to mechanical stress. So basically the shape of our bones and all of the bone markings that you're going to have to learn in lab, they're all there because we have built bone in places where we have stress. So basically we're places where we have a lot of weight bearing on them or muscles pulling on them or something like that. So Wolff's Law states that we grow our bones and remodel our bones in response to demands that we place on them. So when our bones are stressed, the places where we place weight and muscles are pulling on it. The stress is usually off center, so bones tend to bend a little bit. When they bend, the bending compresses one side and it's going to stretch the other side. And when we're talking about the shaft of our long bone, our diaphysis, it's going to be thickest where the bending stresses are the greatest. So where we have the most stress is where we're actually going to go in and remodel and build the bone itself. So here's just an image. It's just showing you that if our bones bend from the stresses we put on them, that one side is bearing the tension and the other side is bearing the compression right there. Other things that Wolff Law explains is what we call handedness. So depending on what hand, if you're right-handed or left-handed, depends on how thick your bones are. So if you're right-handed, your forearm is thicker than the bone of your right hand is thicker than the bones of your left hand. Why? Because we're putting more stress on our right hand if you're right-handed. So the bone remodeling is going to continuously occur here, or maybe we're using our left hand left, so we're not going to have as much stress placed or as much bone buildup or remodeling in that area. When we're talking about curved bones, so like our ribs, those are going to be thickest where they're most likely to buckle. Trabeculate, that's not really important, but the trabeculate spongy bone, how are they formed? Even though it looks like there's no organization, they're actually formed along lines of stress so that we don't buckle our bone. When we have large bony projections, um, where we have heavy active muscles attached, so we're going to build up a lot of bone there. So if we're talking about somebody who's a weightlifter, they're going to have huge bony projections where their muscle attachment sites are to the ones that they're actually using. So when we're talking about a fetus or say somebody who's bedridden, who has no stresses applied to their bones at all, their bones appear almost featureless. So we're not going to see all those bone markings that we described on that table previously because we have no stresses placed on it. We're not being signaled to actually build our bone there. So control of remodeling, just as kind of like a recap, mechanical stresses cause remodeling by producing electrical signals when the bone is deformed. So there's some fluid containing ions. Those are basically the electrical signals. And then this fluid is going to be pushed through those canuliculi so that our osteocytes can go in and be influenced. That's basically the important thing there. This is really important because we talked about hormonal controls and Wolff's Law and mechanical stress. The biggest difference between these two is that hormonal controls determine whether or when remodeling occurs. When we talk about mechanical stress, this is going to determine, hey, where are we actually going to build that bone and remodel that bone?